Good afternoon. This video is about protein purification and characterization. Our learning objectives for this are to be able to develop a plan to purify a protein, tell why proteins are least soluble at their PI or isoelectric point, describe protein characterization methods, describe the theoretical basis of SDS-PAGE, that's sodium dodecyl sulfate polyacrylamide gel electrophoresis. Describe the theoretical basis of mass spectrometry for proteins. Describe the theoretical basis of X-ray crystallography. Protein purification and characterization. We focus primarily on soluble proteins because we can purify those. Proteins such as collagen, that's part of bone, or elastin, that's part of your lung, or keratins, part of your fingernails, are very difficult to purify because they're not soluble. Soluble proteins are therefore easier to study. Pure proteins are easier to study. You can get a lot more really good data from pure proteins. You can't do clean work with dirty proteins. So it's good to have a pure protein to make sure you don't have any interfering activity or interfering protein. So when you measure a function of an enzyme, for example, that protein is all that enzyme and not another one that might be contaminating it. Proteins are drug targets, so once you get a pure protein, you can test drugs against it to see if they inhibit it. Protein purification and characterization. The first thing you need to be able to do is detect and measure, have a detection or measurement method for your protein. If it's an enzyme, you can use an enzyme assay in which you measure substrate disappearance or product appearance. Or if it's a colored protein, you can measure how much of that color is there. Measure total protein. There are various methods for doing that. A real old one is called the Lowry method. The Bradford method is based on Kumase blue. There's also a BCA method that also has a different reagent in it. And then there's an A280 method. It's just absorbance based on tryptophan, tyrosine, and cysteine content. So you choose your methods. You can use solubility, ion exchange, affinity, etc. to possibly purify your protein. We'll talk about those as we go forward. And then finally, once you get it, you can characterize the purity by SDS-PAGE and mass spectrometry to get information on the size of the protein and its sequence. And then you can do amino acid sequence as well by Edman degradation. That's an old method, and mass spectrometry. Edman degradation we'll talk about as well. Determine 3D structure by X-ray crystallography. And now you've got a beautiful picture of your enzyme or protein. Now, proteins fold spontaneously, and that folding is driven by entropy. That entropy is the release of these water molecules and allows them to more freely move about. And the hydrophobic residues in red tend to go to the inside of a protein, and the hydrophilic ones are on the outside. So the water, and over here you have an oil-water interface. That water is not free to move into this space where this hydrophobic residue is. Over here, that water is freed up to move, and that drives the process of entropy to drive the folding reaction. Now, enzyme assays are quite often done in 96-well micro-titer plates or even larger ones, and those are very useful because you can scan wells all at once. You can do a rate reaction on this well over time and compare it to this well that have some standards, and then you can do test inhibitors in various other wells. So it's a really unique, easy way to do it. Most labs have micro-titer plate readers now. There are even some that can read A280 if you have a plate that's UV visible. It doesn't have to be quartz. It can be certain plastics that are UV visible, and you can measure protein in these as well. Centrifugation, you can do differential centrifugation. In various ways, you can isolate nuclear fractions, mitochondrial fractions, or microsomal fractions. You can isolate Golgi and various other things, and that could enrich your sample for the enzyme you wish to purify, and I'll show you an example of that later. You can also dialyze your protein. That is, when it has salt in it, you can put it into a buffer with no salt or water, and eventually whatever the ions are that are small will come out. Your protein, which is larger, will be held in. These bags can be of various molecular size exclusion. Usually it's about 12,000 is standard dialysis tubing, but you can also have various units that use semi-permeable membranes like this under nitrogen pressure, argon pressure to concentrate and dialyze proteins. Proteins are least soluble at their isoelectric point. Proteins have either a net positive charge or a net negative charge, depending on the pH. So at low pH, they'll have a net positive charge. At high pH, they can usually have a net negative charge. And in between somewhere, there will be the isoelectric point, or PI, and that's the point at least charge. Solubility is lowest at this point because there's no repulsion. Over here at positive charge, the molecules repel each other. At negative charge, they repel each other, but at the isoelectric point, there's no repulsion, and they tend to they have hydrophobic residues. They'll stick together. So here's a protein with its hydrophobic interactions, and those can't occur because of the water molecules surrounding them. However, if we want to precipitate a protein, quite often we add a reagent such as ammonium sulfate, which competes with the water molecules. The water molecules are sequestered around the ammonium sulfate, and then the hydrophobic parts of the protein can come together and it precipitates. So protein aggregate via hydrophobic interactions. Anion exchange chromatography is based on charge, so proteins can be positively charged and negatively charged, and this is also a pH-dependent process, as we just talked about. And if you have an anion exchanger, that's a positively charged resin, the negatively charged proteins in blue will bind to it, and the red positively charged proteins will be repulsed and go through and wind up in the buffer wash coming through the column. After the proteins are bound, you wash off anything that won't bind, and then you elute the protein off with chloride. You can actually use a gradient of increasing sodium chloride and separate out multiple negatively charged proteins in the process. So it's a very useful method of separating proteins. Gel filtration separates proteins on size. Large ones go between the beads, and little ones spend time inside the beads. So the little ones come out last. So the big ones come out first, and then the little ones come out last in gel filtration. Now, purification-wise, this is not a very useful technique because a lot of proteins have very similar sizes, and the resolution of this is not great unless you have extremely long columns. But you can use long columns to, you can calibrate them with proteins of known molecular weight and then estimate the molecular weight of your protein. For example, a dimeric protein would elute prior to its monomers. Affinity chromatography is based on the biological function or activity of a protein or enzyme. Proteins lacking the function or affinity do not bind. Examples are antibodies and antigens. So here we have a column that has beads that has an antibody bound to it that recognizes protein recognized by the antibody is red, protein not recognized by the antibody is blue. So all the blue stuff flows right on through, and then the red things stick at pH 7 where the antibody is functioning. You can change the conditions so that the antibody-antigen complex is disrupted. Usually pH 3, like 10th molar HCl works well, and then you can elute your protein over here. You can also put on inhibitors of enzymes onto these beads, and they will bind certain enzymes. For example, you can put a trypsin inhibitor on here and bind trypsin and then elute that at pH 3. Trypsin binds well at pH 7. At pH 3, its binding is disrupted. So you can combine these techniques into multiple ways. So here is an ion exchange profile here with a salt concentration gradient. You can see the activity of your enzyme. The red line is all protein and the activity is in blue. And so you pool these fractions right here and get rid of all the other stuff. Then you run it on gel filtration, and there's some big proteins and some little proteins, and you concentrate on your peak here. And then finally you do an affinity chromatography technique, in which there are lots of proteins that are not bound, and they wash through, and then you change the conditions and elute yours, and hopefully it's pure at this point. So here's an example of a couple of methods for purification of the KEX2 protease that's found in the Golgi of the yeast. Method 1, there was a membrane extraction of lysis of yeast. So you lyse the yeast and extracted out the membranes. You had a crude membrane extract, total protein 736 milligrams. Specific activity 0.38. That's the activity. 280 units of activity divided by the total protein gets specific activity, and we call this 100% yield for the first step, and then enrichment is 1 here. Next time is DEAE cellulose. DEAE has a positive charge on it, so this is anion chromatography, anion exchange chromatography, and that enriches it about 8-fold and increases the specific activity to 3. Benzamidine is an affinity method, and arginine separarosis is an infinity method, and finally we wind up with a specific activity of about 60, and wind up with 1.3 milligrams of protein, and we've purified it 158-fold. Step method 2 involved the isolation of the Golgi by differential centrifugation. Once those were isolated, then they were extracted. Much less protein here, but much higher specific activity to start with. And total activity was about the same. Yield was quite good here relative to this technique. And then DEAE Surfacell is a slightly different reagent, just like DEA cellulose, only the beads are a little different. And then that worked out quite well, high yield there. Then UTI Sephirosis is human urine trypsin inhibitor, and that's hooked on to Sephirosis, and this is a trypsin-like enzyme that likes lysine and arginine substrates. And then, so that one resulted in a real high purity. You got a specific activity of 260, almost 4 times what you had up here, and a real good yield of 72% versus 27% in this method. And so the enrichment factor was 684 here, much higher than here. So this method worked a lot better. Sometimes you have to try various methods to get there. This substrate has a paranitroanilide, so when this peptide bond between the arginine and the paranitroanilide is cleaved, you get this yellow product, and you can measure that in a spectrophotometer, micro-titer plate, and you're good to go. So here's another purification table, and in this one you can see various methods: crude extract and total protein, total phthalate degrading activity. This is units of activity, specific activity, purification fold, and recovery again. And they did ammonium sulfate fractionation here. They just added 0 to 90% ammonium sulfate, precipitated out this activity. Good recovery of activity here, but didn't increase the specific activity a lot, just about doubled it. Then they did a DEAE cellulose, then a CM cellulose. This is a cation exchange technique, and so they wound up with the little better purity. Then they use Cefacryl S200 high resolution, that's gel filtration. That worked well. And then Mono S has a sulfate. It's very negatively charged, and so that worked well as well as a anion exchange, cation exchanger, sorry. And then you got a final pure protein. Now SDS-PAGE. SDS will denature proteins by binding to the protein, and about 1.3 grams of protein bind per gram of 1.3 grams of SDS bind per gram of protein. And SDS is, as I said, is sodium dodecyl sulfate, and so it's a 12-carbon chain with a negative sulfate at one end. So that results in a coated protein that has a constant charge to mass ratio. Doesn't matter what the size of the protein is. Now, slight difference. Some glycoproteins don't bind SDS quite the same as regular as non-glycoproteins. But on electrophoresis, you separate by size rather than by charge, because it has the constant charge mass ratio, just like DNA or RNA. You can also use urea and guanidine hydrochloride to denature proteins by hydrogen bonding. Those are different techniques. SDS-PAGE electrophoresis, you have a polyacrylamide gel which acts as a sieve, and under electrophoresis the protein is negatively charged. So we have the negative cathode up here, and we have the positive anode down here. And since we're moving those anions down, they migrate through here, and then we can the large ones move more slowly because they sieve more slowly through the gel, and the small ones migrate faster, and then you can stain them to visualize your bands. You can also run colored standards, so you can measure approximate sizes here relative to the others that are known. Here's a purification, in which we have a crude extract of a couple of purification steps and final our product here at 40,000 molecular weight. Nice, pure, looks pretty pure. So it's a way of measuring protein purity as we go through a purification procedure. You can also do 2D gel electrophoresis, and this is done a lot in what we call proteomics. And in that process, we do isoelectric focusing in one direction, where we have a gel that with a pH gradient from 4 to 10, and then our protein migrates until it hits its isoelectric point and stops because it has net zero charge at its isoelectric point. Then this gel is turned and put on top of a SDS-PAGE gel soaked in SDS, and then SDS electrophoresis is run. So now we in the second direction, we separate on size. Charge in the first direction and size in the second direction, and you can see how many protein spots you can separate out. You can actually isolate these spots and then treat them with trypsin and then run them through a mass spec and determine what proteins these are. Kind of a neat process, isn't it? That can be used to look at various diseases or different cell proteins in different conditions. Proteolysis is also an important way. As I said, we cleave those up with trypsin. So you need to know what trypsin is. Trypsin cleaves at lysines and arginines, and versus chymotrypsin at hydrophobics and elastase at aliphatic residues. This is the Schechter-Berger Nomenclature for proteolytic enzymes. And here is the polypeptide in black, the bond that gets cleaved in pink here. And so this is the cleavage site. So that peptide bond is going to be hydrolyzed. This P1 residue is the most important residue, usually in proteins. That this would be for trypsin. This would be a lysine or arginine, and then the binding pocket has an aspartic acid with a negative charge that's complementary to the positive charges on the lysine or arginine. That's why it binds well in that binding pocket. But notice that there are other binding pockets as well. There's an S2 binding pocket for residue P2. There's a S3 binding pocket for residue P3, and there's a P1 prime, S1 prime, P2 prime, S2 prime pocket, and P3 prime, S3 prime pocket. So the enzyme, which is in blue, makes multiple contacts with its substrate in black primarily at these different side chain residues, and therefore has specificity is delineated here. Now, so you have trypsin will cleave at lysine and arginine, but if it binds a lot better at P3 and P2, then it's going to cleave faster at if it likes these other subsite binding positions. Kind of an interesting concept, isn't it? So the enzyme doesn't make just one position. This is the most important one, but it's not the only one. Edman degradation sequencing. We can sequence proteins using the Edman chemical reaction, and we use phenyl isothiocyanate to do that, and it reacts with the amino terminus of a peptide. And under the right conditions, this process will cyclize to leave this final product, which is a phenylthiohydantoin amino acid. And these R groups are different, and so we can identify these via HPLC. HPLC is high performance liquid chromatography, also known as high pressure liquid chromatography. And so this process can be done, and you get off the first amino acid, you identify it, and then you do it again with the second amino acid, because you freed up a free amino group here in your peptide when you cleave this off, and then you can do it again. So you move from the N terminus to the C terminus, one amino acid per cycle. Now this is a chemical reaction. It only goes to about 95% completion. So you can't go very far. You can get maybe 10 or 20 amino acids usually, but it was a very powerful technique, and we used it a lot back in many days ago. And I ran one of these brute machines for several years. That was a great experience. Mass spectral analysis to identify proteins. If you can get them to ionize, and sometimes we put proteins on a matrix and hit it with a laser beam to get them to ionize, then they can fly in a vacuum tube, which is called a flight tube, and they will fly based on their charge. So the ones have a higher charge mass ratio will fly faster, and they can be detected. So the lightest ions arrived at the detector first, and the heavier ones arrive later, and so it's called a mass spec. I won't go into it any more than that. However, it's used in proteomics quite often. We have a cell or tissues, we take out the proteins, we run them on gels, we isolate out a band that we think is a very important protein. We can digest that into peptides. We can run that down an HPLC, liquid chromatography peptide separation. Then we can take, as they're coming off the column, we electrospray ionize these things. They give the ionized peptides. The ionized peptides are then analyzed in a mass spectrometer. If the peptide masses resulting from trypsin digestion can be determined by mass spectrometry using peptide mass fingerprinting, this will sometimes allow us to identify what the protein is from that, because we can match this up with the genomic databases and various other databases that explicitly. I'll give you links to that in your handout. But if this does not allow unequivocal identification of the protein via database searching, peptides can be sequenced via tandem mass spectrometry as shown. These peptides then get fragmented again in a mass spec, and then those products, those small ions, get sent on into another mass analyzer, and then we can get the sequences. So the sequence happens to be alanine, glycine, leucine in this peptide. So it's just an example. Now, extinction coefficients of pure proteins. To measure accurately the concentration of pure protein, we'd like to know the extinction coefficient. The extinction coefficient is based on the amount of tryptophan, tyrosine, and cysteine, and of course this formula has been empirically determined, and we like to determine the absorbance of a 0.1% solution. That's 1 gram per mil or 1 milligram per mil, and that at 280 nanometers. So BSA has an extinction coefficient of 0.66, so a 1 milligram per mil solution will have 0.66. HSA is 0.53, and trypsin is 1.57, IgG, immunoglobulin G, is 1.43, for example. Here's the reference for this, Protein Science, back in 1995, and here's a link to the proteomics website, the Exposy website, and we're going to go there, and I will go down to the next slide, and we're going to, go back up. I'll have to escape out of this, and I'm going to copy this amino acid sequence out of here, and then I'm going to go back to this, and I'm going to pull this over in here, and this is the ProtParam tool at the Exposy website. I'm going to paste that sequence right in here. I'm going to hit the compute parameters, and you're going to see that here's the sequence, numbered sequence, 260 amino acids total. Here's the number of amino acids, 260. Molecular weight, 29,253.9. Theoretical isoelectric point is 4.58. This is the composition, amount of arginine, and aspartic acid, glutamic, the amount of arginine and lysine. You can see that you have more aspartic and glutamic, therefore you have the low PI below 7. And then the extinction coefficients, here's the molar extinction coefficient. So a 1 molar solution of this would absorb 280 nanometer light at would have a 46,090 absorbance. You never have a 1 molar solution of a protein. So we deal with absorbance at 0.1%. That's 1 milligram per mil or 1 gram per liter, and that's 1.57. Very similar to trypsin, for example. Actually, this is DNase. And so we have this this is useful information that you can use to understand your protein, what its structure and function is. And so now we're back. And here's an example of a table that has a number of different proteins, and there are different extinction coefficients. And here's the A280 of a 1 milligram per mil solution, or the 0.1% solution. Okay, so useful. Now, X-ray crystallography is once you get a pure protein, you can sometimes crystallize it if you get the solubility just right, and I won't go into the details of that, but you can then hit that with an X-ray beam, and the nucleus of the atoms will diffract those X-rays. Those X-rays can be detected on film, or now we use an instant camera-like method of detecting these, and you get a diffraction pattern from that information on those diffraction patterns. We can use Fourier transform computers and very sophisticated software to create electron density maps, and those electron density maps can be converted into what looks like a 3D atomic model of the protein that's quite useful to study its structure and function. And here are some websites. There are some nice videos for this as well to explain. This is the 100th anniversary of the discovery or development by Doctors Bragg and Bragg, father and son team in England. They developed X-ray crystallography of very small molecules years ago, and now it's been applied to proteins. And here is an example of a structure spinning. This is a model I made, and this is human mast cell kinase, and it has carbohydrate chains stuck on the side. This is the model of a recombinant protein that we made, and you can see the active site serine here, 195, the serine at the bottom of the binding site, 189, the aspartic acid, histidine that are also involved in the active site, and then these carbohydrates are stuck on, one of them to asparagine 95 and the other one to asparagine 72. So it's a very useful method to look at things. Now, Dr. Paul Stanton used to be president of East Tennessee State University, and he was started out life as a surgeon, then a dean, then president. And at graduation, he always gave this quote: "Do all the good you can by all the means you can, in all the ways you can, in all the places you can, at all the times you can, to all the people you can, as long as ever you can." From John Wesley. May grace and peace be yours in abundance.